Assalamu alaikum everyone. Welcome back to a new session in online physiology, and today we will discuss the T cells as the source of cell-mediated immunity. Well, we discussed earlier that acquired immunity is the type of immunity that only develops after exposure to a certain antigen or a disease, and that is antigen specific and disease specific. Now, this type of immunity we call acquired immunity is imparted by two types of specialized cells, the T lymphocytes and the B lymphocytes. Today's lecture is dedicated entirely to the T lymphocytes as the source of cell-mediated immunity. Okay, to begin with, like all other cells in the body, they are produced in the bone marrow. They originate in the bone marrow because we have the lymphoid stem cells residing in the bone marrow. Okay. But unlike other cells which stay there after their birth in the bone marrow, the T lymphocytes move on to the thymus for a process we call pre-processing. And I wish to explain, during pre-processing in the thymus, there are multiple processes occurring. For example, in first place, they rapidly proliferate in the thymus to increase their numbers. Okay? Still later, during their stay in the thymus, they develop reactivity against specific antigens, so that each thymic lymphocyte develops reactivity against a specific antigen. And this process keeps on repeating unless we have thousands of different clones of lymphocytes that are reactive against thousands of different types of antigens. Okay? But during this process, the thymus makes sure that the T lymphocytes, owing to their damaging potential, are not reactive against self-antigens, the antigens that are native to body's own tissues. Okay, and for this purpose, all the T lymphocytes during their stay in the thymus are made to react against a pool of antigens from different body tissues. Okay, and those T cells that are found to react against native antigens or self-antigens are destroyed. They are never released. Okay. In fact, over 90% of the T lymphocytes processed in the thymus are destroyed before release. So this is how the thymus takes care of safety. Safety is above all. Okay? And there is one more thing. The prefix T lymphocyte probably derives because they are processed in the thymus. T for thymus, T for T lymphocytes. Still later, after their pre-processing in the thymus, they move to their storage sites. They are extensively stored in the lymph nodes along the blood vessels in the body. Okay, but in addition to the lymph nodes, they are also stored in the thymus and the tonsils, in the adenoids, in the spleen, and the gastrointestinal tract, in specific pockets of lymphoid tissue, and in the bone marrow. So perhaps they are stored in every part of the body. They are stored advantageously. And that storage is strategic as well. Here I wish to explain what I mean by this term strategic. Okay, they guard the portals of entry into the body, so that any microbe or antigen that can enter the body through the nostrils or through the pharynx, oropharynx, will be intercepted by the T lymphocytes residing in the tonsils and adenoids. Okay, if a certain disease microbe our antigen is smart enough to escape that level of protection, that is filtered at some other level. For example, if it enters the body through contaminated food and water, the pockets of lymphoid tissue in the gastrointestinal tract, that is the Macrophages, will have those T lymphocytes which will intercept the invading microbe. Still later, if it is even smarter and somehow manage to enter the bloodstream, the T lymphocytes and the spleen, thymus, and bone marrow will intercept that organism and limit its spread in the body. Okay, so you need to remember their storage is not only advantageous but strategic as well. Still later, now here they are a bit weak. Like many other cells in the body, like the neutrophils, the macrophages, and the B lymphocytes, which are stimulated or activated directly by an invading microbe or an antigen. The T cells are dependent upon cells we call antigen-presenting cells in the body for their activation. They don't get activated directly by the antigen. Rather, they need help from the antigen-presenting cells in the body. And as we mentioned in previous classes, we have three types of such antigen-presenting cells: the macrophages, the B-lymphocytes, and the dendritic cells. Okay, if they ask you to choose one, they give you all these options in a question. They ask you to choose one. Go for dendritic cells because they are the most potent of all the antigen presenting cells. Perhaps antigen presentation is their only function. Okay, and how this process occurs, let me show you. This is an antigen presenting cell, any one of these. Most of these are phagocytic cells. After phagocytosis of the invading microbe, what they do, they break it down into smaller fragments or pieces, and those fragments are displayed on the surface of antigen presenting cells. In association with certain proteins we call MHC-proteins. They are coded by genes we call major histocompatibility complex. So they need to be presented on the surface of antigen presenting cells in association with MHC proteins to be identified by T cells and for them to be activated. Okay. Here I wish to add, there are two types of MHC proteins. We have MHC1 class 1 proteins and we have MHC class 2 proteins. Okay, and those are specific for activation of specific T cells. For example, an antigen presented in association with class 1 MHC proteins will only activate the cytotoxic T cells and not the helper T cells. The helper T cells will be only activated if the specific antigen is presented by the antigen presenting cells in association with class 2 MHC proteins. So that too needs to be taken into account. Okay. Once they are activated, what they do next, we will cover in next slide. But one thing is important. Each time they are activated by a specific antigen, they do what they have to do. But in addition, a clone of those T lymphocytes activated by a certain antigen proliferates to give birth to more and more T lymphocytes to combat against that antigen, but a certain fraction, that is a certain fraction of them goes back to their storage sites and stays dormant over there. Okay, and we call them memory T cells. And the significance is during their first exposure to a certain antigen, they get the technical know-how to combat it, how to fix it, how to eliminate it. And now they give rise to the memory T cells that stay dormant in their storage sites on re-exposure to the same antigen, not any other antigen. Those memory T cells are activated. Okay, since they already have the experience and technical know-how to tackle that specific antigen, so the second time there is exposure, the response on part of the T cells is quicker, rapid, within days. Okay, and it is very potent, unlike the first exposure. Okay, so the memory T cells serve to offer a rapid response, a quicker response, and a potent response on re-exposure to a certain antigen. That's all about them. Now we move on to the specific types.